Final. Okay, so we'll continue from there. Just note down these things. In the last class, we have already seen what's the principle behind this generation. So, what we have understood is this: whether it is neurons or in muscles, which we have seen in the earlier chapter, they can undergo some transient changes. There will be rapid fluctuations in their membrane potential. So, we've understood that initially the membrane potential will be the way how it is shown here. You can see outside positive, inside negative. This is how the membrane potential is, which is going to undergo some fluctuations. Okay, this condition is what we call polarized, which is going to be depolarized. Right. And again, it will undergo change back into the polarized condition, which we are going to call repolarization, repolarized. So these are the changes that we are going to see now. Okay, so just note down this statement. These things you have already noted down, right? Leakage channels as well as voltage gated channels are clear, right? Okay. So then just write a figure like this. I mean, a table like this. So what we are going to see here is condition of leakage ion channels and then voltage gated channels. So what kind of changes they will be undergoing? The first column is membrane potential. So here we are going to write membrane potentials. On the other side, what is the condition of leakage ion channels? What is the condition of voltage gated channels? Okay, so in this, leakage ion channels are always open. So this is by default you'll write open only. During resting membrane potential, if you just see this thing, initially Na plus voltage gated channels as well as K plus voltage gated channels are closed only. Okay, so what is the condition of this membrane is resting membrane potential. So during resting membrane potential, that is polarized condition. What is the condition of various channels means? Must see this Na plus K plus pump is operative. It is already transporting Na plus and K plus in the opposite directions. This is already taking place. There are more K plus here and negatively charged proteins here, more Na plus here. Coming to the channels, voltage gated channels are still closed only. At that time, what you see is membrane potential, that is resting membrane potential. That's the first condition. Okay. Now, upon stimulation, which channels are going to open first? Hmm. So, sodium voltage gated channels will open. So, this is what the first thing that happens when the axon is stimulated. So, when these are open, as we know, what happens is there is influx of Na plus. So, Na plus will be entering inside now. That is influx. Na plus influx. So, with this, what happens is depolarization, which is what we call action potential or now impulses. What is going to happen is this. This opening of Na plus voltage gated channels is actually for a shorter period only. So after that, what is happening here? Na plus voltage gated channels are closed, immediately followed by opening of K plus voltage gated channels. So what happens here? There will be a flux. Sorry, there will be a flux of K plus. So K plus is leaving. So when K plus is leaving, what happens to the charge again? It is repolarized. Just write about the conduction. There is no much discussion about the conduction of nerve impulse in the textbook, but what actually happens is this: when you take point A, which is in the depolarized condition, point B, some point I am taking, which is still in the polarized condition. See, as we know, uh, in B plus, that is in the B region, there are Na plus voltage gated channels. The same way, there are K plus voltage gated channels. So this one will act as the stimulus for this, right? Which is voltage, of course. This point already having voltage. What is that? Depolarized condition, which will act as the stimulus for the next point. So therefore, what happens? Na plus voltage gated channels will open. So whatever we have discussed for A here, same thing happen with the B. Only thing is the stimulus is this point now. Is it clear? Okay. So later, by the time it is stimulated, undergoing depolarization, this one is again repolarized. Okay, so the same way from B to C, C to D like this, along the axon, nerve impulse is getting conducted. So instead of this giving this explanation, I'm just giving you the textbook one. Just to write this line, this one. This line is about conduction. Completed? Okay. Next, right? Saltation. Saltation or saltatory conduction. Right. Here, the logic we need to understand is, see, let us take action potential is in point A. Right. At this point, it is in the depolarized condition. This one will act as a stimulus for B, right, which is going to undergo depolarization now, which means the action potential is basically passing like this in smaller circuits from B to C, C to D, and so on. Right. Now, just if you take the same thing here, if this is the point where action potential is present in this point, right? If I take point B, but point B means just a region which is close to this, it is already covered with what? Myelin sheath. As we know, this myelin sheath is actually insulator. It is insulating one because it is fatty layer basically, it is insulator. Therefore, here action potential cannot generate. In this point, action potential cannot generate. What is action potential again? The changes which are happening across the membrane. For the action potential to generate, Na plus has to be transported into the axoplasm. All these changes should happen across the membrane. This membrane part, I'm just highlighting only that particular part. See, this part of the membrane is now completely can't be involved in the exchange part. This part of the membrane cannot undergo depolarization. No polarization also. Is this logic clear? Because it is insulating region. Okay. Therefore, if action potential is present at this point, at point A, right, where it can stimulate this next action potential? Here. Got it? From here, this one will act as similar, means current will be flowing from this point to this point and so on. So what is the difference between this unmyelinated or non-myelinated and myelinated one is, here the conduction is taking place in smaller circuits. Smaller circuits means what? From this point to this point, this point. So in a smaller circuit, this conduction is taking place. But here there is a jump in the conduction from one point, this point to this point. There is a jump. Right. When we say saltation, the meaning of saltation is actually jump. Even in evolution, we use the same term. When we say saltation, technically the meaning of this thing is jump. That is jumping. So what is jumping here? Action potential is jumping from one node of Ranvier to the next node of Ranvier. So which conduction will be faster? Understood the logic, right? Which one should be faster now? Right? This one will be faster. So conduction of nerve impulse will be faster in the myelinated axon than in the non-myelinated one. Understood? So most of the neurons are myelinated only. So therefore there will be faster conduction. To start this point. So what is the importance of myelin sheath means? It will be helping in the faster conduction of nerve impulse. Okay, and if you are clear with this, what if the myelin sheath is, for example, present throughout it without any node of Ranvier? What if it is present? There is no node of Ranvier. Will this conduction of nerve impulse means? The answer is no. No conduction here. Why no conduction? Because this point, for example, if there is action potential, this cannot stimulate this point. The distance is too high. Okay, so for this action potential to jump, there must be a specific distance that is required. That is what you will find naturally among the node of Ranviers. So if the question is about this, what if the neuron is completely, that is the axon is completely covered with the myelin sheath means no conduction in this. That. So we have discussed about generation, generation of nerve impulse, and then conduction. Now, conduction means how it is conducting in the axon is what the discussion is. The next one is transmission. See, this one is a simple logic. Let us take this is the axon endis. The axon terminal is this. Okay, so let us take the next membrane which is present or contact with this or to which this nerve impulse has to be transmitted. Let us take it is in very close proximity. It's like they're very close, so they're almost in contact. So in that case, what happens is whatever this action potential present in this membrane will directly jump into this, which means action potential present in this can stimulate this membrane. Now, when this can be possible, when these two membranes, this membrane and this membrane are in close contact. Is it clear? So whatever happened in the conduction part, that will happen in this transmission part also. This kind of synapse is what we call electric synapse. right? But if the distance is this, that is, if there is a space between these two, there is some kind of distance between these two. Here, the action potential cannot directly jump into this. Okay, so instead, what happens is inside this axon terminal, this one is axon terminal. This is the synaptic nobis. I mean, let me call this one is particularly this particular part. This is pre-synaptic membrane, whereas this one is the post-synaptic membrane. Right. Why? Because here you can see the formation of junction. This complete junction is what we call synapse. A synapse, wherever we come across, the meaning is junction. So this synapse consists of presynaptic membrane, postsynaptic membrane. In between these two, whatever this space is there, this is what we call synaptic cleft. Cleft means basically there is a space. So now how this action potential can be transmitted from this to this means here comes the role of neurotransmitters. So inside this axonal knob, inside this knob, there are vesicles. These vesicles are filled with chemicals called neurotransmitters. So basically what happens here is when the action potential reaches this point, it will stimulate these synaptic vesicles to go and fuse with this membrane. It's like exocytosis basically. Right. So this is the presynaptic membrane is, and the vesicle is this. What vesicle is this? Synaptic vesicle. This vesicle will go and fuse with this membrane. So whatever the neurotransmitters present here are going to be released inside this. Let me show you. After fusion, this is how it is. The contents are released here. After this, what is there? Postsynaptic membrane. This one is the postsynaptic membrane. In the postsynaptic membrane, there are receptors. Any? Okay, so these neurotransmitters will go and bind with this. Right, see in the discussion that we were having, I told you there are two different types of voltage gated channels, ligand gated channels and voltage gated channels. Okay, so what this membrane is going to have is, what do you call this one now? This one consists of ligand gated channels. So when the neurotransmitter bind here, it will stimulate these ligand gated channels. The same thing, there will be influx of Na plus, the same story. But only thing is the stimulus is different. Stimulus is ligand. What ligand is that? The neurotransmitter binding with the receptor. Is this clear, everyone? Yes or no? Right. See, just for the idea, I'll just show you how this channels will be inside this. Okay, I'll show you that before the just copy this first. What do you call this synapse, by the way? That's it. Okay, chemical synapse. You can draw from the textbook better so that it will be a practice. Just a second. This one. So here, what you see is presynaptic membrane, postsynaptic membrane. In between, whatever the cleft is there, synaptic cleft. So this is completely what we call synapse. So in between, neurotransmitters are released, going to bind with the receptors. So action potential which is present in this membrane is now going to jump into this. Okay, so inside this membrane, what you find is ligand gated channels. So draw from the textbook. Manish. Okay. Okay. So just write an additional note which can be useful. Conduction of nerve impulse. hmm, Conduction of nerve impulse is unidirectional. Now means what? When you take axon, one side is cyton, the other side is axon terminal. Is it clear? Okay, so what is the conduction of nerve impulse? Only this. Okay, so just think about it. For example, now listen everyone. For example, I am just taking 3 points here again. A, B, C. When B is in the depolarized condition, action potential is present in this point. Okay. From here, the action potential is passed to A or C. From B, action potential is passed to B, A or C. will be passed to C, not back to A, right? What the A is? A is in the repolarized condition. No? This one C is in the polarized condition. Okay, but actually, technically speaking, repolarized condition, polarized conditions are same only, right? The only thing is it is once again polarized. This one is already in the polarized condition. So both are in the polarized condition only. So can the action potential pass from B to A? Can this stimulate again A? Means according to this statement, no, it is not. It is unidirectional. It will pass only from A to B, B to C, and so on. Is it clear? Okay. Why? What is the reason for this thing? Means why it cannot be conducted back from B to A means a small additional concept we need to understand. I'm just giving you that may be useful. Right, that is this. Okay, see properly here. This is the graphical representation of what we have discussed. What was happening there? Just see one by one. Uh, what happened? Is it visible? Okay. Now see properly. On this y-axis, you see membrane potential. Okay, in millivolts, membrane potential is shown on the y-axis. On the x-axis, you take time. Now, initially, what do you see here? This is baseline. Minus 70 millivolts is what the resting membrane potential is. Understood? So minus means what we have seen is inside is negative, outside is positive. That is resting membrane potential. So this is resting membrane potential. Okay, upon stimulation, say this is the stimulus. So when the stimulation happened, what happened to this impulse is, now we see from negative it is changing towards the positive. So initially, actually this is what we call threshold. Threshold potential, which is around minus 10 to 15 is what it is. So once this threshold potential is provided, now you can see a green color line. It is undergoing what? Depolarization. So from polarized to depolarized. How do we depolarize to? Plus 30 is what the membrane potential is. So inside it is becoming positive, right? It is depolarization is. This is depolarization phase. Is it clear up to this? Okay. After that, again it is changing back into what? Minus 70. This phase is what we call the repolarization phase, which is shown in the orange color here. So this one is a repolarization phase. See, when the repolarization is taking place, there will be a case called hyperpolarization also for some time. Okay, but again it is converted into polarized resting membrane potential only. Right, now in this, if you see this particular part, I'll just highlight in the red in this. Refractory period, relative on the absolute refractory period. So what do you see here? These two colors, observe properly in the figure, means this part. Now, so from this point to this point, I'm talking about this. In the first phase, it is in the depolarization condition. Second one, it is in the repolarization condition. Okay. And next one is this color. What color is this? This one, all this. So basically, from this point to this point, if you just see overall, it is what we call the refractory period. In refractory period, again, there are two phases are there, but basically, let us take it as together. It is the refractory period. So from where to where? From this point to this point, it is in the refractory period. What is meant by this refractory period is when the membrane is in the refractory period, it cannot be further stimulated. Understood? It cannot undergo depolarization or whatever it is. So stimulation cannot be possible if it is in this phase. Again, once it reaches this phase, after this, if there is a stimulus, yes, there is going to be again the same story. But before this, if stimulus is provided in any stage of this thing, there is not going to be any kind of depolarization. If you are clear with this logic, now think about it. Now you have 3 points now on the membrane. A, B, C. If B is in the depolarized condition, that is action potential is present in the B point. A is already in what phase? Refractory phase. Because it is already undergone stimulation, right? From this point only, already action potential passed to this. So A is in the refractory period, either this or this refractory period. So B cannot stimulate A. Got the logic, right? Therefore, the now impulse will be always in one direction. But this one is, if you see the conduction part in very depth, I don't think that this much depth question will be asked, but still. So what you write here is, here, why there is, means now impulse is conducted in one direction. Reason, what reason we can write? Refractory period simply. If at all this one has given as assertion, the reason for this thing is what? Refractory period. If you want to write, you can write like refractory period means what? It is a period during which further stimulation means further stimulation cannot be possible. Everyone? Okay. Next. Excitatory versus inhibitory postsynaptic potential. Okay, so here the logic is this, must see properly, simple logic. Presynaptic membrane, postsynaptic membrane. Postsynaptic membrane, that synaptal cleft. Here, neurotransmitter is released from this. This neurotransmitter is going to bind with the receptor here. Okay. Once it binds with the receptor, what will be the change that is going to happen in this membrane? Is it polarized or depolarized? It is already in the polarized condition. Upon stimulation, it will undergo depolarization, right? This is what we call the action potential, right? Understood. But same presynaptic membrane, postsynaptic membrane. There are some neurotransmitters released from the presynaptic membrane that makes this membrane undergo hyperpolarization. So membrane, instead of undergoing depolarization, undergoing what? Hyperpolar. It is already in the polarized condition. It is undergoing hyperpolarized condition. Means, for example, here minus 70 millivolts was the resting membrane potential. Yes or no? If it is changing to plus 30, usually this is what it is. What do we call this one? Depolarization. But here, minus 70 millivolts will become, say, for example, minus 90 millivolts. It is already in the polarized condition, undergoing further polarization. This is hyperpolarized. So you can clearly see that the first one is excitatory. This is excitatory. Means what? It is actually stimulating the next membrane. But here it is inhibitory, right? A depolarized condition is excited condition or activated condition. But instead of undergoing depolarization, it has undergone hyperpolarization. Is this logic clear, everyone? Right. This is what we call inhibitory postsynaptic potential. Inhibitory postsynaptic potential. This one is, first one is excitatory postsynaptic potential. EPSP. This one is IPSP. So, what is the importance in the biological system is, it is not always activation of the next membrane. Sometimes inhibition is required. For example, you are studying about this sympathetic, parasympathetic. Right? One is stimulating, the other one is inhibiting. So, inhibition is required as well as activation is required for the control. Is this logic clear? So, how that can be possible means, this is the story. Now, what makes the difference here? What kind of neurotransmitter is released? So, whether it is going to be excitatory or inhibitory, it depends on neurotransmitter. What are the neurotransmitters that you know? Now, give me one example, sir. Acetylcholine, good. For example, acetylcholine, if you see, it is actually excitatory. Now, what is mentioned in the textbook is only this, excitatory one. But if you see gamma aminobutyric acid, in short, we call this one GABA. It is also a neurotransmitter. This one is actually inhibitor. Is it clear? Right. If no questions, copy it neatly. This is also an additional concept which is required. So the point is, in the biological system, both are required. Activation is required, inhibition is also required, which you have already seen in that discussion on this ANS. In the biological system, most of the synapses are chemical synapses. Why? What is the advantage of chemical synapse over electric synapse? Must see, now I have already given the answer. Problem is we are not able to correlate it. Okay, what is the advantage? See, whenever something is discussed, you need to understand the significance of it, advantage of it. So chemical synapses are more in number. Mostly we have chemical synapses only. Electrical synapses are very few in number. In fact, which one will be faster? Conduction. It is faster. Electric synapse is faster, right? How does it help? It is not about the question and answer. First of all, few of you are not even trying to think about it. But the thinking ability is what is lagging, which you have to work on. I don't know why you are not getting. Shubhalekha, are you trying or waiting for Kanishka to think? If you are not thinking means you have decided that you are not capable. You have decided it. Yes or no, Asif? Think man, don't depend on the others because you have to write your own exam. No, Kanishka, come on. I've already given the answer. This ability you have to develop, Kanishka. I told you now. I've already given the answer. There is no regulation. So that's what the discussion we had. So whether the next membrane has to be stimulated or inhibited can be possible only in the chemical synapse. This discussion is about the chemical synapse only. I need not to tell it separately. You can see that depending on the neurotransmitter only, whether excitation or inhibition is taking place. In electrical synapse, it is not possible. Okay, right. We'll stop here. In the next class, we'll discuss about brain and its functions. Go through up to this. Hmm. What happens to? No, no. That is not actually related to action potential generated across the membrane. That cannot stimulate the action potential. Okay, actually, that you have to take like this, not just electric shock or something. There are receptors. Which receptors we have? The mechanoreceptors. So, in the electric shock, electricity is directly passed among the cells. That is a different kind of conduction. Here, what the discussion is about the conduction that related to polarization, depolarization. For example, when there is electric shock, whatever, for example, the pain is there, that is happening through these neurons. That is what this action potential and conduction is about. No, no, no. Actually, the term we call now is that, but it is actually blood vessels. What is blood vessels? Superficially, what are present are veins. Nerve injection. What injection? Mm, yes. The, eh, no, which surgery? In the sense, you're talking about the anesthesia. No, local anesthesias and these anesthesias will work on this principle that they will inhibit the conduction of nerve impulse. Two things here. One is there are some drugs, right? Not only that, low temperature also. Few drugs under uh, low temperature. It is cold, for example, if you see ice, they will decrease the speed of conduction. Like if you have some pain, what you'll do is immediately you'll keep the ice, no? So it will relieve the pain because low temperature will decrease the conduction of nerve impulse, speed of conduction. The same way, there are some drugs which will also decrease the speed of conduction. They are used as like local anesthesiants. Like procaine is one drug is there. So there are some drugs. So drugs means local anesthetics and low temperature ice decrease the speed of conduction is what it is. 34. Phosphorylation means what phosphorylation is there in the plant in the chloroplast? What kind of phosphorylation is there? It is photophosphorylation only, right? So enzymes or whatever these factors related to photophosphorylation is present in the quantasomes only. Quantasomes means what? They are uh, all the pigments along with this ATP and all related to capture of light energy. They will form units called quantasomes. Here, quanta means light, means unit of light. So, quantasome related to units of light absorption, which are related to this phosphorylation. They are present in what? Thylakoid membrane. So, basically, pigments only. Clear? 146. The thin filament of skeletal muscle fiber, a small globular protein, masks the active sites on F actin. See, the question is small globular protein. Okay, as we know, let me just show you that. See, tropomyosin is actually filamentous one. It is not globular protein. Globular protein is this. Okay, tropomyosin is not the globular protein. So directly it is globular protein, but who is helping in this blocking of this thing is troponins. So it is actually NCRT line also. Here. In the resting phase, subunits of troponin mask the active binding sites of myosin. These are actually globular proteins. So it is troponin. Why it is not tropomyosin? Are you clear? Tropomyosin actually directly blocks, but it is actually filamentous one. It is not the globular one. And also NCRT line. This one. It does not mean that in light reactions there are no enzymes. Light reactions also have enzymes, but very few. See that photosynthesis last one. Effect of temperature in the factors. Go there once. So see that you'll find a statement saying that biosynthetic phase involves more enzymes than light reactions. It does not mean that no enzymes in light reactions. Got it? Right. Any other doubts? Clear all? No. Right. So prepare after this. In the tomorrow's class, we'll see brain. And for now, we don't discuss that reflex action and the uh, this sensory organs. As of now, they are not mentioned in the syllabus. If required, we'll see later. Otherwise, we'll just see the brain part alone, and then we'll move to chemical coordination. You have a older new textbook. Okay, so as per the new syllabus, we have only till brain. The spinal cord and this reflex action and all not there in the syllabus. Okay, right then. Thank you. Bye.